Good morning, Honorable Vice Chancellor, Dean School of Life Science, Senior Professor G. Guru Subramaniam, and dear participants. So I welcome you all on the behalf of Mizoram University and my own behalf also. Basically, this webinar series has started by our Honorable Vice Chancellor. He is the brainchild behind all this webinar. And in this pandemic situation, Mizoram University has been continuing the disseminating the knowledge of this webinar series. In past also, we have conducted many webinars, and future also we have different plans for webinar also. So in this line, today we'll have two talks. The first talk will be given by Dr. Gabrielle Britton. Topic is Aging and Health, a New Initiative. And second talk will be given by Dr. Vivillarreal. And topic is Translating Research for Neurodegeneration, Challenges, Biomarker in Neurodegenerative Disorders. So first, let me introduce the first speaker, Dr. Gabrielle Britton. After going through her CV, I think if I start talking her CV, it will take one lecture. So very brief, I am giving the introduction of Dr. Britton. She has completed BA in Psychology from Vanderbilt University and MA in Behavioral Psychology and PhD in Neuroscience and Psychology from Indiana University in Bloomington. And after going through her research, we find that she is basically working on the finding the biomarkers for the age-related problems, basically the Alzheimer's. And in same line also, another speaker, Dr. Vivillarreal, also his specialization is biotechnology, molecular biology, biochemistry, immunology, and basically neuroscience, cognitive aging, neurodegenerative diseases. And also his research is basically based on the finding biomarkers for early diagnosis of age-related problems. And so with this brief introduction, first I invite Dr. Britton to continue her talk. So Dr. Britton, please.
Thank you. Let me share my screen. Can you see my screen?
Yo la puedo ver, sí, sí se ve.
Okay. Okay. Good evening, everyone. Thank you for this invitation. It's a privilege to share with you tonight our research. Dr. Villarreal and I have been working together for almost 10 years now, and we're very happy to join you tonight in this webinar series. I'm going to begin by giving a background on the program that we developed together over the course of the last 10 years, and give you an idea of the initiatives that we have in place in aging research in Panama. First, to begin with, I'd like to draw your attention to the changing demographics around the globe. As you know, life expectancy is increasing in virtually all countries. Very few countries have not had significant increases in life expectancy. And one of the regions where the aged population is growing very rapidly is in Latin America. So if you can see my screen here, since 1990, only 7.3% of our population was aged 60 years or older. And by 2050, that percentage is expected to be 25%. So 1 in 4 people will be aged 60 years or older in 2050. And these increases in population aging are certainly indications that we have, it's a success story, essentially, that more people are living longer because people are surviving early childhood and vaccination programs have been very successful. And at the same time, people are living longer in their adult years. But the population aging also brings with it many challenges. And I have summarize these challenges in 3 different categories. They're very broad categories, and they're not exclusive categories. I think they overlap in many ways. The first of these, which is our primary interest in our working group, is the challenges related to health. So as populations age, there are more people in our different countries with age-related chronic illnesses, which are very narrowly associated with aging. And at the same time, a lack of specialized services, or I should say an insufficient amount of specialized services, and specifically gerontology and geriatrics. And this is true across the globe. And statistics show that very few people are are going into geriatric specialties where, in fact, many more specialized physicians are required. Another general category in which countries are being challenged is economically, as we know that if there's the number of working people is decreasing versus the number of retired people is increasing. And then we know that there could be relatively more people claiming pension benefits versus people working. And so this puts pressure on a shrinking workforce to increase taxes, for instance. So these are challenges that we all have, all our countries share. There are also social challenges, of course, in caregiving. We need more caregivers. A lot of this burden is carried by women in different families, especially in Latin America. And so altogether, we have many challenges to face in population aging. What I'm showing you in this graph here is that particularly associated with the challenges in aging and health. So what you can see in this graph is the percentage of cases across the different age groups. This is from our the Bureau of Statistics in Panama. This shows like the disease burden in our country. And what you can see here is that in very early childhood, if people die very early in childhood, typically it's due to infectious diseases in this blue part of the graph here. But as we live longer, as we survive through the, in our 20s and our 30s, most of us are going to suffer from and eventually die from chronic illnesses. So it's this red line here. So even though Panama is a tropical country, I think there's a mistaken idea that most of the diseases that are prevalent in Panama are tropical diseases, when in fact they're not. The top 10 diseases that are responsible for mortality in Panama, only one is an infectious disease, and that's HIV, which has practically become a chronic illness now. So a particular illness that, or a particular condition that is very narrowly associated with aging is dementia. And here in this slide, what you're looking at is it's a very good description of how dementia is an umbrella term that describes a collection of symptoms caused by disorders that affect the brain. So it's not one specific disease. It's how thinking is affected and behavior and ability to perform everyday tasks. And dementia is very narrowly associated with aging. And so as populations age, we also have more and more cases of dementia. The most common form of dementia is Alzheimer's disease, followed by vascular dementias. And of course, there are other types of dementias like Lewy body, frontotemporal dementias, and other dementias associated with Parkinson's disease. If we look at dementia statistics, these are very important, especially in countries like Panama and even countries like India. We know that right now there's about 50 million people. I think in the latest concrete statistic was 2017, 50 million people around the globe living with dementia. And this number is expected to double every 20 years. So you can imagine the huge burden this has on health systems. 2 out of 3 cases are in low to middle income countries. And in fact, the 2 countries where increases are greatest are in China and in India. And in Latin America also, but the 2 greatest countries are China and India. And altogether, dementia accounts for $800 billion in cost. So 1% of global gross domestic product, which is a huge amount of money. In fact, a very common statistic that is cited is that if dementia were a country, it would be the 18th largest economy in the world. And you can see in this graph, for instance, that as dementia cases increase, that the great majority of them are going to be in low and middle income countries. So the cases in our countries, like in Panama and in India, are growing at a faster rate than in high income countries. The costs of dementia, as I said, are $800 billion in costs. And people tend to think that these costs are associated with medical costs, when in fact, 40% of the costs are related to informal care, and 40% to the social costs, the cost of people having to stay at home and losing their jobs because they have to care for somebody, or because there aren't enough systems in place to care for people with dementia. Only 20% are accounted by medical costs. Another interesting aspect of Alzheimer's disease is that over the course of the past few years, and the last 2 decades, in fact, many important diseases have been able to be decreased, their prevalence and the number of deaths, because of novel medications and new technologies that have permitted people to battle these diseases. For example, breast cancer and prostate cancer and heart disease, stroke, and HIV/AIDS. And this contrasts very starkly with Alzheimer's disease, where actually there's been an increase in number of deaths. So we have a very challenging situation with Alzheimer's disease. In fact, also the burden of Alzheimer's disease is also carried disproportionately by women, where as we age, women have a chance, 1 in 6 chance of developing Alzheimer's versus 1 in 11. And this sex difference between the development of Alzheimer's is not solely due to women living longer, although it's not really clear what it's due to. It's probably related to differences in hormone systems. But this is also interesting because Alzheimer's is a disease that disproportionately affects women also, because women are also disproportionately the ones who are the caregivers of people living with dementia. Another important challenge of Alzheimer's disease is that it is the disease with the highest rate of failure in pharmaceutical development. And this creates a situation where in the last few years, there have been no new drugs that have been approved in clinical trials. And many of these drugs pass preclinical trials, but then they fail in phase 2. In fact, every single morning we wake up to different journal reports on failed clinical trials. For instance, these are just from the past couple of months. In one case, a particular drug, minocycline, was not a candidate for patients with symptomatic Alzheimer's disease. And in the other case, another drug was also not well tolerated and was also failed. And we see lines like this often in the literature, that these are expensive, painful, and fruitless investments in drug development. So it's very disheartening. We have huge challenges in front of us. So that's a little bit of background on the challenges we're facing in terms of population aging, combined with the difficulties in developing novel drugs. Now I'd like to discuss a little bit about why the disease is so challenging from a clinical perspective. So we know, for instance, that Alzheimer's disease is a very complex disease. I think that sometimes in popular literature, we tend to say that Alzheimer's disease is about the loss of memory alone, but actually it's a lot more complicated than that. It has various different types of disturbances. For instance, cognitive function is severely impaired, and it's not only memory that's impaired, but also spatial and temporal orientation. There's language disturbance. There's a difficulty identifying objects. And there's also neuropsychiatric impairments, for instance, mood disturbances and agitation, aggression. I'll show you other examples. And there's also a very important functional component. So as the individuals begin to progress in the disease, it manifests a reduced ability to carry out their everyday activities, for example, being able to dress or handle their money or take care of themselves. So these are all very challenging for the individuals and also for the people they live with to be able to carry on everyday life once the illness begins to progress. In this particular scheme, you can see all the different types of clinical symptoms that emerge as the disease progresses. And what's interesting about the clinical signs of Alzheimer's disease is that they express themselves in different proportions depending on the individual. So it's also a very heterogeneous disease, and doesn't express itself in the very same way in every single person. There's impairments in judgment and withdrawal and personality changes and difficulties to complete tasks that all tend to progress as the disease progresses. In terms of cognitive function, we see, for instance, that over the course of aging, even under normal circumstances, various cognitive processes are affected. In this particular graph, you can see that most cognitive processes decrease normally with age, such as perceptual speed and verbal memory, and others have a more, they flatten out more, for instance, like verbal ability or just general knowledge, numeric ability, for instance. These also tend to flatten out. But when Alzheimer's disease interferes, these decreases that we see here at later ages tend to be much more pronounced. So dementia takes a toll on individuals in terms of their cognitive function. In this particular graph, which I thought was a very interesting summary of not only does Alzheimer's disease manifest itself in cognitive impairment, but also in neuropsychiatric impairment. So what you're looking at here, for instance, is the number of neuropsychiatric impairments that emerge even before diagnosis. So before Alzheimer's is diagnosed, we're already seeing some neuropsychiatric impairments like disturbances in sleep and depression, and there's suicidal ideation and some anxiety. And once the diagnosis is made, other neuropsychiatric symptoms emerge. So this should give you a general idea how complicated this particular disease is. Once, for instance, the disease progresses a lot more, we see that individuals have difficulty feeding themselves or taking care of themselves and dressing and being able to use the bathroom on their own. These are basic activities of daily living, as we refer to them. They're primary outcome variables in many of our aging studies. The other primary outcome variables, the instrumental activity of daily living. These are also activities that we carry out in our daily life, like being able to go out into a restaurant, for example, or go shopping or do our finances. These are also impaired. In fact, these are impaired before the basic activities are impaired because they have a stronger cognitive component. The others impair later. So how is Alzheimer's disease diagnosed? Well, what I just described to you is a number of different impairments that make up the disease. So in the case that an individual should require an evaluation, essentially it's a very complex evaluation that involves taking a clinical history from the patient and family member, a physical evaluation where we learn about their chronic illnesses, take their blood pressure, etc. The cognitive status, of course, is one of the most important because it's a strong factor of how the brain is working at that particular point in time. A depression screen is essential to rule out the possibility that cognitive dysfunction is due to depression. There's a functional assessment, how well the person is able to carry out their daily life. And if possible, the introduction of biochemical biomarkers, for instance, in cerebrospinal fluid, and imaging biomarkers. And I have these in blue because these are not readily available at all in all clinical sites, especially, for instance, in primary care. So these would be more common in primary care, especially a very brief cognitive screen, nothing too pronounced, but these are less available. So to summarize, one of the things that we know in particular about Alzheimer's disease is that all the changes that occur in the brain begin almost 20 years before the symptoms appear. So keep this in mind. This is very important. It has a lot to do with the work that we conduct as Alzheimer's researchers. So all the symptoms appear 20 years after the changes in the brain begin to occur. And these symptoms emerge as a consequence of neurodegeneration in brain areas that underlie cognitive function. So the brain starts to deteriorate far before, many years before, like the cognitive symptoms appear. And imaging tools complement the clinical diagnosis, like I said, but they're not readily available. So we have an urgency to develop different types of tools that can help us diagnose Alzheimer's before symptoms appear, but that are readily available in countries, especially like Panama and even India, in all countries. So in this particular graph, what you're looking at here is an illustration of the pathologies, the neuropathologies associated with Alzheimer's disease, and when they appear in relation to the symptoms. So here's the age of the individual, and you can see the symptoms typically appear much later, after the neuronal atrophy occurs. So the symptoms appear much later in the course of the disease progression. And the very first changes that occur are the deposition of a protein called beta amyloid beta, and then an inflammatory response to this accumulation of amyloid beta, followed by the phosphorylation of tau protein, which produces tangles in the neuron bodies and causes the integrity of the neuron to suffer, and so the neuron falls apart. But the important thing is that all these changes in the brain are occurring 2 decades before the symptoms appear. So the goal of all Alzheimer's researchers is to be able to develop tools that allow us to identify individuals in this particular phase before the symptoms appear. So that if there could be an intervention, whether it would be pharmaceutical or behavioral or both, it would have an opportunity to be able to at least retard the disease or slow it down. So to summarize, if we look at the progression of cognitive function from a normal individual to, for example, a state of dementia, what we're looking at here is an aged individual who has normal development could progress to what we call mild cognitive impairment, which is a phase that precedes dementia. So a certain proportion of older adults develop mild cognitive impairment, or MCI, without progressing to dementia. But a certain percentage of these do progress to dementia, and they could spend up to 10 years, for instance, in a state of impairment. It requires caregiving, et cetera, before passing away. So this is very challenging. Our goal is to be able to identify people at this phase so that we could intervene in some way before they progress to more severe stages of dementia. So just to give you an idea, it's estimated that the annual rate of conversion to Alzheimer's in persons with mild cognitive impairment is about 10 to 15% per year. So that's the annual rate of conversion from MCI to AD. So the importance of biomarkers should be evident at this point. For instance, in our region, in Central America, estimates suggest that 90% of cases of dementia are diagnosed in later stages. So 90% is a huge number. This is very common in Panama, where neurologists and other specialists don't actually see an individual until they're already very much advanced in an advanced stage of dementia. And the principal barrier to diagnosing AD is the lack of tools to identify the disease in early stages. This is true across all countries, and especially in low to middle income countries like ours. So this brings me to where Dr. Villarreal and I co-founded a group that we call the Panama Aging Research Initiative. And we've been working together with many different specialists in different fields, and psychologists and neuropsychologists and different physicians and biotechnologists, chemists, etc. And we have started the first longitudinal study of aging in all of Central America, actually. So we are the only ongoing longitudinal study of aging in the region. And the reason why we began the study was to be able to work together on a problem that's common to all countries. So aging is a factor that's affecting, it's a global challenge. And no one in Panama, no research group was focused on that until we began to look at this problem. And just recently, Dr. Villarreal first authored this paper in which we published our protocol. So you can find this online in Medic Review, where we published our study protocol. Our protocol was approved by 2 different bioethics committees in the country, and all our participants signed informed consents as in accordance with Declaration of Helsinki. Just to summarize a bit what our program is what our objectives are. Our general objective is to characterize cognitive function in elderly adults in Panama using a longitudinal approach. And the specific problem that we're looking at is that age is the primary risk factor associated with MCI and Alzheimer's disease. The size of the problem is that we know that AD prevalence is increasing, that AD diagnosis is challenging, and that this also has a very strong sex component, where women represent 65% of AD cases and 80% of caregivers in the region. So we have a very huge challenge. Let me tell you a little bit about the types of variables that we measure in our longitudinal study. So we collect demographic information, clinical, we have clinical data that we collect, like the number of chronic illnesses and how many different types of medications adults are taking, whether they're experiencing depressive symptoms, how they evaluate their quality of life. We do a really extensive and in-depth cognitive assessment. We measure frailty, and then we also take a blood sample. And I'll tell you a little bit about that, but Dr. Villarreal will talk a lot more about that when he gives his talk. By the way, this protocol, the way that you see it illustrated here, is very common across all aging studies. So running a longitudinal aging study is not about deciding individually what we would like to look at, but we basically adopt a very standard protocol that's used across all different research groups that are interested in aging. And this is especially important so that we can compare our data across cohorts. So here's a summary of the different interventions that we do. And over the course of time, since 2011, we have repeatedly tested subjects. Some of them are with us since the very first assessment, some of them since the second assessment, and all the ones that are active right now have been with us at least since the third assessment. And I'll tell you a little bit about these two different cohorts in just a second. So we have been able to run two different cohorts since 2011. I'll tell you a little bit about these because it's very interesting how different they are. One is a clinical cohort. When we first began, we conducted our study in an outpatient geriatric service in the largest public hospital in Panama, in the city of Panama. And as the years progressed, we then collected a community cohort. And you see that the first cohort had over 400 people, the second has over 300 people. And they're different in a number of different ways. So here we're looking at the first cohort at the left of each graph. And the top graph, we're looking at the age difference of each cohort. So our first cohort, the median age was 80 years of age, 80 years of age. And we collected data from individuals from 65 years and older. And our oldest participant was 102 years old. So a very old cohort, a very aged cohort. 80 years was the median and the mean as well. And in our second cohort, our community cohort was much younger. We began recruiting now age individuals 60 years and older. And the mean age of our second cohort is roughly 68 years of age. So a younger cohort, the community cohort is younger. In terms of years of education, this first cohort, this age cohort, the clinical cohort, had a very low level of education. In fact, the average years of education was 7.1 years. So a great number of the cohort, a large proportion of people in the cohort had not finished primary school if the average was 7 years. Whereas our community cohort, the average years of education is roughly 16 years. So one of the advantages that we have of having two distinctly different cohorts, as you can see here, is that we can then study aging from a number of different perspectives. As we know that age is a significant factor that influences the way that we age. So our first cohort was much older, so we have the advantage of being able to examine very aged individuals. We also know that education is a strong risk factor, low levels of education for dementia. So we can look at the data from the perspective of people with low levels of education versus higher levels of education. So these are the two cohorts that we're working with. Our current cohort is this community cohort. Typically in any type of longitudinal study, what we're looking at is a number of different factors, not just risk factors, but also protective factors. So you're looking at the top half of this diagram, you can see the protective factors that are associated with aging, what makes us age well and be less susceptible to dementia, versus the risk factors, the factors that make us predisposed to developing dementia. And all of these are interesting in an aging study, especially a longitudinal study, because our aim essentially is to be able to identify in our population the particular risk and protective factors that are associated with dementia. So that's the overall goal of our program and the work that we conduct. So for instance, the interviews that we conduct get at a lot of this information. We'd like to know what factors our Panamanians have that could protect them against dementia. And then we also collect information about unhealthy behaviors like alcohol use, smoking, unhealthy diets, and these different chronic illnesses that predispose to dementia. So some of these are, you know, obviously modifiable behaviors, like whether you smoke or whether you have unhealthy diet or you abuse alcohol, that predispose also to these different chronic illnesses. But some of these risk factors are non-modifiable, like our genes, for instance, right? And these are not modifiable, and the passage of time is also not modifiable. So these are things that we have in common. And just looking at this particular graph should give you an idea, this particular figure should give you an idea of the complexity of Alzheimer's disease. So like I said in the beginning, it's a heterogeneous disease with a number of different risk factors, and you can arrive at Alzheimer's disease from a number of different pathways. So we know, for instance, that depression is a strong risk factor for Alzheimer's disease, and inflammatory responses, as well as, you know, metabolic factors, oxidative stress, and different lipid pathways are associated with AD. And so probably the causes of Alzheimer's are multifactorial, and not a single pathway is involved in producing AD, and probably is also the reason why it has been so difficult to develop any type of a pharmacological or some type of, you know, agent that can possibly interfere with the deterioration associated with Alzheimer's disease. This complexity is what probably makes it so challenging to find a suitable medication that can mitigate the symptoms. So one of the first publications that we had that we published together was characterizing a small cohort of Panamanians with Alzheimer's disease and mild cognitive impairment in our cohort. So there was a subgroup of people who were identified by consensus diagnosis, and in which we looked at different factors where we identified, for instance, that age is a risk factor, as we know, for Alzheimer's disease and MCI, but also really importantly, the apolipoprotein E4, which is one of the genes that Dr. Villarreal is going to talk to you a little bit more about. ApoE4 is one of the strongest genetic risk factors for sporadic Alzheimer's disease. And we were interested in identifying, first of all, if this was a risk factor in our population, because different findings have been reported for different populations. And we did find that it is a risk factor. So having 1 or 2 copies increases the odds of having Alzheimer's by 5. Here's the 95% confidence interval. So anywhere from 2 times to 13 times more likely to develop MCI or Alzheimer's if you have 1 or 2 copies of the E4 allele. And Dr. Villarreal is going to talk a lot more about this, but ApoE4 plays a pretty strong role in development of Alzheimer's because we know it's involved in the production of amyloid beta anomalies, and individuals with 1 or more of these E4 alleles are more likely to develop Alzheimer's. Other papers that we've published have looked at some of the behavioral factors associated with aging. I'm going to review some of these for you to give you an idea of what we're doing. In this particular table, what I've done is I've summarized some of the factors associated with cognitive impairment, with symptoms of depression, or with both of these together in our cohort. So as would be expected, we know that age is a strong risk factor for impairment. And it turns out it predisposes people to having both cognitive impairment and depression at least 3 times more. These are odds ratios. A low level of education increases the risk of all of these conditions across the board. So having less than 6 years of education, not having finished elementary school, that first primary school, predisposes people to cognitive impairment and depression in later life. This is very important in populations like Panama, where the average age of the population, the education in the population is about 9 years. So it's very critical to increase years of education because this is a modifiable risk factor. So it's the responsibility of governments to invest in education to reduce the risk factors that are associated with low levels of education. Having more than 4 chronic illnesses also predisposes individuals to having depression or to have both cognition and depression impairments. And of course, difficulties in basic activities of daily living and instrumental activities of daily living are narrowly associated with cognitive impairment. And these were published in a series of papers that we published a few years back by Dr. Villarreal was the first author. We also looked at another outcome variable, which is very common in aging studies, which is falling. As we age, one of the risks that increases is the risk of falling because our muscles get weaker and also our balance is affected. So falls are a really important outcome variable in aging studies because they are indicative of potential risk of hospitalization and early mortality, for instance. And so what we looked at is that we looked across our cohort and looked at the different risk factors associated with a single fall in the last 12 months versus multiple falls, more than one fall, for instance. And one of the interesting findings here is that we find that depressive symptoms were associated with multiple falls. So this is something that we can look out for in the clinical environment. And interestingly also is a couple of inflammatory biomarkers, I309 and IL-7. Both of these increase the risk of multiple falls and of single falls. I'll tell you a little bit more about these biomarkers. So over the past 7 years, Dr. Villarreal and I and our team of researchers, we have started collaborating with a group of researchers around the world and primarily in the United States, looking for blood-based biomarkers in Alzheimer's disease. And Dr. Villarreal is going to elaborate on this much more, but to give you an idea, one of the reasons why many groups are focused on the search for blood-based biomarkers is because drawing blood is a very easy procedure in primary care. And in fact, any community clinic, people can draw blood. And so it could be a really, really important step forward if we could somehow use that opportunity to develop a biomarker that can be readily available more widely than the imaging biomarkers that are already available, like MRI or PET scans, et cetera. And so many different groups are focused on looking for blood-based biomarkers. And we are working together with all the people you see in this list of authors, and most of them are based in the United States, and looking for blood-based biomarkers so that we can try to test the idea that blood-based biomarkers could be one way to identify Alzheimer's in early stages. And this is our primary collaborator in the United States, Dr. Sid O'Brien. He's at the University of North Texas Health Science Center. This is Dr. Villarreal many years ago, and our other colleague, Dr. Carrera, who together we work on, we measure a number of different blood-based biomarkers. And so some of these are very bad for aging, so bad, some of them are like inflammatory, acute and inflammatory chronic biomarkers. Some of these are growth factors that we measure. Some of these are anti-inflammatories, and altogether there are a number of different, as you know, that there's many different proteins in blood. It's a very complex fluid biological system. So one of the first things that we did was to see if a combination of proteins measured in blood could somehow distinguish between Alzheimer's disease patients and normals, or MCI participants and normals. And what we find here is that this is a Gini plot. What it does is it ranks the importance of the different proteins and how readily they can distinguish between one group and another. And these are the top 21 proteins that distinguish AD persons from normal controls, and the top 21 proteins that distinguish MCI from controls. And as you can see, many of these are the AD and MCI have in common. So many of them are inflammatory proteins. And the other thing to notice is that none of these are the classic proteins involved in Alzheimer's, which are amyloid beta and tau pathologies, for instance. So these are novel proteins that can possibly indicate also novel pathways and implicated in Alzheimer's disease. Why is this useful? What is useful about what we're doing so far, and where are we at this point in time? Well, with our same collaborators, what we did is that we pooled all our protein data and our blood protein data to see whether to increase the power of our data, to see how well these blood-based proteins could distinguish between normals and controls, and sorry, between ADs and controls and between MCIs and controls. And one of the most interesting things that came out of this analysis is that the combination of proteins was very good at telling us who did not have AD and who did not have MCI. So very high negative predictive value in each case, 95% in the AD case and 94% in the MCI case. And why is this important? Why could it be useful? Why would it be useful that a combination of proteins in blood would have a high negative predictive value? Well, as it turns out, the majority of screening tests that are used in primary care and other settings have a very high negative predictive value. So they're used to rule out people who do not have a particular pathology, who then need to go on and get further tests done. For instance, in tests of depression or prostate cancer or mammograms. Those initial tests that are conducted are important in order to rule out whether you have a particular condition, and are actually have a high negative predictive value. And if something doesn't turn out right in this particular test, you're recommended to go on and get a more specific test. And so what it does, it could be a way to screen in primary care people who do not have a particular pathology. And for instance, we propose in this paper that we published together that the blood screen could be used in primary care, and anybody who would be a potential or a possible case of Alzheimer's, because it has a high negative predictive value, we can rule out everybody who doesn't have, who's not at risk for Alzheimer's, could then go on and get the rest of the tests that are more time consuming, that cost more money, and that are less available in primary care. So would go on to neuropsychological tests, for instance, and to different types of imaging tests and different specialists. So in primary care, it'd be very useful to have a test for blood-based biomarkers, where we can rule out people who were not at risk for a particular disease, as in other diseases. So the blood-based screen could be very useful in this way. And this is where we are at this point. So one of the things that I mentioned in the beginning was how Alzheimer's is not a curable disease so far. But I wanted to draw your attention to a publication that came out just a couple of years ago and is still being talked about pretty much. And it basically is a meta-analysis that was done by the Lancet Commission in Dementia, where what they looked at, for instance, was the percentage of risk that's due to different types of risk factors over the course of life, and how these add up, and whether they're potentially modifiable. And so if you look at something like low education, it accounts for 11% of the risk in Alzheimer's. So if we could eliminate, or if we could improve our education systems across different countries, we could definitely mitigate Alzheimer's by quite a huge number. 11% is a huge number. And if we add up all these different modifiable factors, in the case of Latin America, they add up to 56% of risk factors that are responsible for dementia are potentially modifiable. 56% means that if we could tackle smoking, physical inactivity, for instance, prevalence of diabetes and other factors, we could reduce dementia by 1 out of 2 cases, which is a whole lot of people. And these data were similar for other low middle income countries. 1 out of 2 dementia cases could be diminished if we tackled modifiable risk factors. So while we find some medication that could possibly mitigate Alzheimer's disease or be able to deal with the symptoms, there's a huge potential to mitigate Alzheimer's through modifiable risk factors. While we try to identify a better way to diagnose Alzheimer's in early stages, one approach is definitely to encourage older adults during their middle age to have a healthier lifestyle, so that aging isn't burdened with these risk factors at a time when the brain is particularly susceptible to neurodegeneration. And this has been clearly pointed out by this intensive clinical trial called FINGER, which is very interesting because it's an intensive a clinical trial, intensive intervention, a very complex intervention that involves nutrition, counseling, physical activity interventions, cognitive training, and management of a number of different risk factors. And it has been, it's hugely expensive, it's very time consuming, but it basically is testing this hypothesis that we can intervene extensively in late middle age to mitigate the, the cognitive effects of aging and neurodegeneration. And you can see clearly here in the red line here, the ones who received the intervention and their total cognitive score is much higher, significantly higher than a non-intervention group. And in several different domains also, these individuals showed improvement. So this is a very well-known clinical trial. It's called FINGER. You can look it up in Google. There are several different papers that have been published. And this is just one example of how intervention in modifiable risk factors does have the potential to reduce the prevalence of dementia in later life. Finally, I'd like to just end with a general statement about some of the challenges that all of us are facing. The first of these is that we need more data in order to have an impact on aging age populations. So we need to be able to understand these patterns, and so we can tackle disease and disability burden. This is also important for identifying age-appropriate allocation of resources. Right now, for instance, in the COVID epidemic, this is more true than ever. It's more evident than ever that in the initial stages of the pandemic, we were given the impression that older adults were more severely affected by COVID than younger adults, and many countries were not prepared to deal with this because older adults are already isolated and are already a vulnerable population. So it's very important to be able to identify what their needs are, what the chronic illnesses are, what sorts of challenges they have in order to allocate resources appropriately. And finally, the most important thing is to be able to develop prevention and intervention approaches in order to minimize the disability associated with aging. And one of our main goals as a group is to be able to, eventually develop clinical trials that are not non-pharmacological, but are behavioral interventions, as it's similar to the FINGER trial, that can mitigate the impact of aging in our population. Thank you. I'd like to thank many different groups of people have helped us. I wanted to be brief. I'll leave my talk there and let Dr. Alcibiades continue. And I'd like to thank all the people on this list. As you can see, we had a very important collaborator in India, the Acharya Nagarjuna University, where we first met Dr. Sambasiva Rao, and this is where we met, where we know him from. And these are our other partners. Thank you very much.
Thank you, Dr. Britton. And now we are going to conduct a question and answer session that will be handled by my colleague, Professor G. Guru Subramaniam. Before starting this session, I want to make an announcement that participants who want certificate kindly write to this event organizers, that is Professor S.K. Mehta or Dr. Vikas Kumar Roy. So we will send their participation certificate within a few days. So now I am just requesting Guru Subramaniam to conduct.
Thank you. It's a very nice presentation. Really,
Thank you.
I'd like to thank you in an elaborate way. Because of your experience, you explained in a detailed way. Even I don't know anything, but I learned many things from your presentation. It's a very good to see you. So now it's a question answer session. So now I request all the participants to post their questions for this session immediately. So that way the speaker can answer your questions. The first question is, Madam, is Parkinsonism an age-related disorder or any other causes?
That's a good question. Yes, Parkinson's develops earlier than Alzheimer's disease, but it does get worse with age. So it is an age-related disorder because it develops later in it. So very few young people get it, and as we age, we're more likely to get it. So it is age-related, and it's all neurodegenerative disorders have something in common, which is some kind of a protein malfunction in the brain, and either accumulation or malfunction, and so and this also increases with age. So
Thank you. The second question is there any protocol recommended or authenticated by any organization for Alzheimer's disease?
What type of protocol? You mean like diagnostic protocol or research protocol?
Okay. No.
I don't. What type of protocol? What do you mean?
It is based upon the diagnostic part.
Yeah. There's a guideline. There are guidelines for Alzheimer's diagnosis that are based on the National Institutes of Aging, and it's essentially they involve, you know, a full clinical. You have to meet the clinical diagnosis of certain cognitive domains have to be affected, and your activities of daily living have to be affected enough to interfere with you being able to carry out your daily life. And at the same time, that is coupled with the presence of at least one biological marker, whether it's detected by imaging or in cerebral spinal fluid or another type of marker. So the combination of those two things, the cognitive impairment, the impairment in activities of daily living, and at least one biological marker.
Thank you, ma'am. The next question is aging also a programmed cell death? Aging?
Well, wow, that could be an entire seminar. We're not really sure why we age. That's a really big question in biology, why we age. But no, I don't think it's programmed cell death. No.
The next question is there any role of yoga or pranayama in the prevention of aging?
Well, that's also very interesting. I know that there are clinical trials that have been conducted that have shown, for instance, that people who practice yoga and meditation and those types of that has a very positive influence on cognitive function and on decreasing depression, and probably has also an impact on development of Alzheimer's disease, although I haven't seen like widespread clinical trials based on that. I wouldn't be surprised that that would be the case. Probably people would still develop Alzheimer's, but much later. So any type of intervention where you have more physical activity and you're physically healthier, you have some type of a cognitive reserve that even if you develop Alzheimer's, you develop it later. So you have sort of a period where you're buffered from that ramping up of cognitive impairment that occurs when you're first diagnosed.
See, there is an announcement. Hello. There is an announcement from the organizing committee side. Professor Baby is there. Please come.
Yes, sir.
Yes, you can
yes,
ask.
Can you
questions.
hear me?
Yeah, you may ask
questions
to the speaker.
All right. Yeah, okay. This is, head of Department of Psychology in Mizoram University, and I'm also General Secretary of the Alzheimer's and Related Disorders Society of India, the Mizoram chapter.
Nice to meet you.
I'm very, very glad that you have brought these latest information to us. Thank you so much, first of all. And
thank you for inviting us.
I'll try to make it quick. My question is on the personality factors that changes way before memory losses and other neuropsychiatric or functional and other cognitive factors are apparent. I am in my group, we are over 100 Alzheimer's caregivers. Even my mother and my friends, mostly moms, you're very right. The rate is very much higher in women in the meso population as well. And, you know, what all of us have, you know, noticed when we share our experiences is the personality changes that happens way before these other cognitive and neuropsychiatric and functional, you know, symptoms appear. You know, each family would be able to report when a specific personality change happens. Say, for example, faulty perceptions about, you know, about relationships with closed ones, things like that, you know, blaming other
Sure.
people, you know, all of these things happen, we feel, in a very early stage. You know, in your presentation, you hardly spoke about personality changes. In the longitudinal study that you are conducting now, personality, is that included in the cognitive aspect? I don't know. We're very interested in studying this personality or highlighting the personality aspect of this illness. Is there a research methodology? Because, you know, can this be studied retrospectively? Is there a good, you know, research methodology that you could suggest other than longitudinal study?
Well,
Yeah,
those are lots of questions, and I agree.
Yes.
It's very interesting. And one of the slides that I did show was a study that was published in Alzheimer's and Dementia, where
Yes.
you know they do talk about neuropsychiatric changes, but if you look at the data, like some of these changes occur before diagnosis, and some of them include things like social withdrawal or accusatory behavior, which could be considered, of course, changes in personality. We're not really measuring things like that in our protocol,
Okay.
but I guess I'd have to look back into the actual protocol to see if there are particular items that do have those characteristics. But I agree with you. I think that those are you know, interesting, and the thing is difficult to measure because probably there's so much variability between different people, you know. And as we see, sometimes some people become more aggressive, other people become more withdrawn, but certainly those personality changes precede the diagnosis. And I agree with you, ma'am. I think it's very interesting, and I don't know if there's a particular protocol that is established for that. But what I can do is I can send you that particular citation from Alzheimer's and Dementia.
Yes, please.
Because it looks like they measured a bunch of different neuropsychiatric conditions. Thank you very much for the question.
Thank you so much. Thank you so much.
Thank you, Professor Baby. Yeah, this is the question from Anuradha David. What are the underlying genetic mechanisms of aging?
Oof. Underlying genetic mechanisms of aging. There are several different genes involved in aging. In fact, they've been identified in anywhere from the same genes in invertebrates and vertebrates. But the interesting thing is that in some systems, some basic systems, the genes are turned on and are associated with aging. In other systems, the genes are turned off and are associated with aging. But there are a set of different genes, you know, that have been identified that have to do with longevity, if that's what we're discussing. But the direction of their expression differs from, you know, whether they're, you know, more primitive organisms and more complex organisms. But Dr. Villarreal is going to talk about many different genes involved in neurodegenerative disorders that I think are also, that can be, you know, narrowly associated with aging processes as well. So I think I'll leave that for his presentation so that he can get started as well.
Thank you, ma'am. This is a question from Telagavadi Subaya. Is there any recommended dietary pattern for the prevention of Alzheimer's disease?
I think that healthy aging, you know, whether we're talking about neurodegenerative diseases or just healthy aging in general, I think they converge upon, like, you know, having a good level of physical activity and low levels of saturated fats and high levels of, you know, omega-3, omega-6, and eating fish oil, that what we call the Mediterranean diet. So I think that it's not just exclusive to neurodegenerative diseases, but that it has been shown that these all reduce chronic illnesses, they reduce obesity, and also keep your mind more active, and keep, you know, your brain works better if you are not burdened with all these chronic illnesses. And so I think that good health, good eating, and good activity converge upon healthy aging, independently of what different, you know, condition we're looking at aging.
This is the question from Victor Das. Have you narrowed down to one potential biomarker to diagnose Alzheimer's disease?
No. No, we haven't, and nobody has. It's not going to be a one biomarker thing, and it's not going to be a one target, one protein. It's not going. It's too complicated for that. Too complex.
This is a question from Madhavi. Is there any relationship between cervical dystonia and Alzheimer's disease? If yes, what is the possibility?
I don't know the answer to that question. I don't know.
Then another question from Asha. Do we have any diagnostic reports of Alzheimer's in young children?
No, I don't think so. Maybe not Alzheimer's, but dementia, as I said, is an umbrella term that is a loss of function, and, you know, that can be caused, for example, by brain trauma, or it could be caused by, you know, HIV, for instance, HIV-associated dementia. So you can suffer a really bad blow to the head and have a version of dementia, and but not Alzheimer's in childhood, no.
Okay, thank you.
However, there are Alzheimer's cases that are have a genetic basis that you can develop in your 30s, which is still pretty young to be in your 30s and have Alzheimer's dementia, for instance. There are cases of that.
Thank you, ma'am. This is the question from Dr. Mukesh Ranjan. Why sexual differences exist in Alzheimer's disease? How?
Well, that's a very, yeah.
Yeah,
that's
please.
an interesting question that's, you know, widely studied. It appears to be related to the fact that women undergo a loss of estrogen at a point in their lives where, you know, like when we go through menopause, and the impact that may have on neuronal function. And so estrogen is neurotrophic, and it stimulates neurons. And so those changes that we undergo as women over the course of development appear to have an effect on a later life, although it's not really clear in what direction. So it may have something to do with when we go through menopause, or even if we undergo childbirth or not, for instance. So there are many different groups working on this, and the only thing that's clear is that there is a sex difference, and it's not entirely related to the fact that women live longer. There is a sex difference in AD.
See, there is a lot of questions from the participant. So now at the end of the session, we will continue that.
Well, you could also, you know, you could email me the questions if you like, or, I mean, I don't want to take into the time of Dr. Villarreal.
Okay, I will do that. I will do that. Okay. Thank you, Doctor.
Thank you.
Thank you, Dr. Britton.
Thank you, Dr. Rao.
Thank you.
So now we are going to start second session of today's talk. That will be given by Dr. E.Villarreal, and the topic of this talk is translating research for neurodegeneration, challenges and biomarkers in neurodegenerative disorder. So now I am requesting Dr. E. Villarreal to give his lecture. So please.
Yeah, I'm trying to share the screen. Okay, let me see. Do you see it?
Yep.
Okay. So thank you very much for the invitation. It's a great pleasure to me to share this panel with Dr. Britton, and also to see people like Dr. Sambasiva that I've been seeing for a couple of years. So thank you very much. As you already mentioned, I work with Dr. Britton in Paddy Research in Panama, and the focus of our research mainly is the search for biomarkers in neurodegenerative disease. So as an introduction, as a brief introduction, I have to mention that the main reason that we focus our research in the search of biomarkers is because we are trying to identify an early diagnosis and risk assessment, as already Dr. Britton mentioned very well. So we are trying to look biomarkers that we can use as diagnosis, monitoring disease progression, and also risk assessment. It's very important to mention that all the biomarkers can be used as a complement to clinical assessment. And in the case for Alzheimer's disease, we are using clinical symptoms and at least one of 3 biomarkers to try to diagnose Alzheimer's. And the biomarkers that has been approved so far are the PET imaging, MRI imaging, and CSF proteins. In the case of the PARI study, that our focus has been the search of CSF proteins, ApoE genotyping, and of course, blood-based biomarkers, as we already mentioned. It's very complex when you are looking biomarkers for neurodegenerative disease because you have so many points that you can look for different biomarkers. In this slide, I try to show you that we have amyloid pathology, you can have tau pathology, synaptic degeneration, alpha-synuclein pathology, glial activation, neuroinflammation, TD43 pathology, that is the marker for Parkinson's disease that you've been asking so far, prion pathology, and neural injury. So the neuron, microglia cells, astrocytes, all part of the cells in the neuron can be affected by different type of proteins or malfunction of proteins. In the case of amyloid pathology, you can have accumulation of amyloid beta 42, amyloid beta 40, amyloid beta 38, and amyloid beta oligomers. In the case of tau pathology, you can have total tau that can be in the body of the neuron, and also you can have phosphorylated tau as a form of neurofibrillary tangles. These neurofibrillary tangles, what it does is that they disrupt the structure of the neuron and they cause the death of the cell. Also, you can have synaptic degeneration by different proteins, neurogranin, NPTX2, alpha-synuclein inclusion, and this is for glial activation, very important, and neuroinflammation. A protein that has been very studied is the protein TREM2, also the YKL40, and also a lot of cytokines, as we've been seeing, that cannot only be detected in the brain, as you can detect it in the blood. So what is very important is that when you are studying biomarkers, you have to not only see the point of view of the protein and how you can detect it, also you have to relate it with a lot of health assessment in the elderly. For example, you have to do health assessment. You have to see if this individual has chronic disease, cardiovascular health, to see his weight, to see his height, muscle strength, mental rating, as I already mentioned in the talk of Dr. Britton, cognitive function, as memory, attention, concentration, affective state of depression, anxiety, and functional status. Something that has been the focus of our research, and the last year the World Health Organization set the guideline, is that we are trying to see how we can risk reduction of the cognitive decline and dementia. So to try to understand how the biomarkers affect the Alzheimer's disease, we will see the evolution of the diagnostic criteria for Alzheimer's disease. In 1984, the biomarkers, the only way that we can see it was by biopsy or autopsy after death. In 2011, these biomarkers, as amyloid plaques and neurofibrillary tangle, was included, and you can be, and also besides doing by autopsy or biopsy, you also can do it by different methodology as PET imaging, MRI imaging, and CSF protein. And what the revised diagnostic criteria said that you could have 3 stages of the disease: preclinical stage, mild cognitive impairment, or Alzheimer's disease. So now, so far in 2018, these biomarkers have been updated, and also you can have a system of that you can rate the disease as if you have amyloid beta levels in CSF, you have tau levels in CSF and also PET, and if you have neurodegeneration as but PET imaging and structural MRI. So if you want to try to see to understand how you can conceptualize the major stage of dementia or Alzheimer's disease, you will have 2 stages: preclinical state of Alzheimer's disease and the clinical states. So in time, you can see that you can have amyloid beta depositions so many years before that you have the clinical symptoms. Once you have the clinical symptoms, your brain has been accumulating this amyloid beta deposition and tau pathology for so many years, and you will have cognitive impairment, and in this point you will have positive molecular biomarker for Alzheimer's disease, as for example amyloid beta 42 and tau protein and phosphorylated tau protein. And a couple of years after you show the first signs of the cognitive impairment, you will have an impact in your daily activities, and you can characterize as Alzheimer's disease, and you will have a typical Alzheimer's disease, typical and mixed dementia also. So this is a way to see the impact that have these proteins that we've been talking about as amyloid beta and tau in the brain. As in this picture you have a normal brain, and in the other way you have a brain with Alzheimer's disease. As you can see, this space over here, the ventricles are more affected because of the memory loss, and this has been due to the accumulation of these amyloid beta proteins and tau proteins. In this graph, you can see the biomarker change in a logarithmic scale, and in the beginnings, and how is this change becomes in the disease progression. For example, in this, for example, in the case of CSF amyloid beta, you can see that it starts and at the very beginning to increase. After the amyloid beta can be detected in CSF, after that you can detect it in PET imaging. There you can detect the tau protein in CSF. After that you can detect MRI and glucose PET by imaging, and then you will have the impairment. But before the impairment, you can detect already all these protein by different methodologies as PET imaging, MRI imaging, and CSF measurement. You will have a broad range where you will have a loss of synaptic, and after that you will find that at the very end. So when we talk about genetic biomarkers, it's very important that you also measure the clinical phenotype. In the way that right now we are measuring the genetic biomarker for Alzheimer's disease is that we are measuring a lot of SNPs to try to see or to understand different disease pathway, because as Dr. Britton mentioned, we are not against a very simple disease that you can measure one biomarker and you can define this disease pathway. You have to measure a lot of things to try to figure out this disease pathway. After you do the disease pathway, you will have a gene co-expression. You can have different expression of gene that can coexist at the same time and can go different pathways. And this expression in the brain, we will have these networks that you need a lot of bioinformatic tools to try to understand what is happening with these genes. I like this slide because it shows you that for different type of cells in the brain, you can have different genes that you can target to try to understand what is happening in the neuron, what is happening in the astrocytes, in the endothelial cells, in the microglia, in the immature oligodendrocytes. So for every cells in the brain, you will have different genes that you can target to try to understand and define what are the pathways that are involved that leads to this neurodegeneration. In this graph, you will have all the genes that so far has been targeted for Alzheimer's disease. There are so many different genes that you can see, but what is very important is that you will have 3 specific genes that are penicillin 1, penicillin 2, and amyloid beta precursor protein gene that if you have one of these 3 genes, you will have autosomal dominant Alzheimer's disease gene. And it become expressed in 100% of the individuals. These cases are the familial Alzheimer's case, and most of them express very early in life, from 30 years and go on, and they don't live longer than 50 years in most of the cases. But you will have, and what you see is they are expressed in very low frequency, but they have a huge effect size in the case of Alzheimer's. But these are at least between 1 and 5% of all the cases of Alzheimer's disease. It's very low. That's why they say that it's very infrequent, or this low frequency of expression. But you will have a lot of genes in this range over here that you will have more frequent, but the effect size is lower than these autosomal dominant genes. I put in the red boxes the gene that we are trying to target in our research, because has been found that has a good effect size and also have a good frequency of appearance in the population. For example, this is a neuronation sequencing panel from Illumina that we are targeting to try to understand genes for Alzheimer's disease, Parkinson's disease, ALS, frontotemporal dementia, dystonia dementia, early onset dementia, and dementia from Lewy body. So you can see, it's so complex to try to figure out the mechanisms and the underlying pathways for Alzheimer's disease that you have to measure all these genes over here to try to figure out what is happening with the cells within the brain of people with Alzheimer's disease. The same thing is for Parkinson, and most of the genes that you have for Alzheimer's also are in Parkinson and ALS and frontotemporal dementia. They are not excluding for one disease, so you can find the similar pathway within one disease and the other one. So what we are doing as a precision medicine approach for evaluating risk of dementia is that we have two groups. We have people with cognitive impairment, that is at least 95% of all the people that will develop dementia, and people without cognitive impairment. So you will have to take in account clinical assessment for both of them. And what we are doing right now is trying to do genotyping, whole exome sequencing for them. That is the previous slide that I already mentioned. And you will try to detect which known mutation you can identify. If you identify any of them, you can say that you have a diagnostic confirmation, and you can do individualized management plan for typical symptom and disease-specific complication. And you can consider this group for considering to enrollment in a clinical trial. If you have identification of a gene and that is known mutation for Alzheimer's disease, and in this group, you will have a very high risk for future dementia. In the case that you don't have any known mutation, you can target for ApoE4 carrier. And this ApoE4 carrier, that's, as we already explained, I'm going to talk a little bit more further, increase the diagnostic likelihood that you will have appear Alzheimer's disease in both of the case. But it's very important that all this data, you have to use clinical longitudinal trials to do electronic integration with biomarkers, genetic data, genomic public annotation to identify which one of these groups could be involved in development of dementia. Now we are going to talk a little bit more about the apolipoprotein E. In the case that we want to study, we'll focus our attention in the apolipoprotein 4 alleles because it's the one who confer the greatest genetic risk factor for Alzheimer's disease, for late onset Alzheimer's disease. This protein plays many important roles in the transport of cholesterol and lipids in the brain, and also exists as tripolymorphic alleles, E3, E4, E2, E3, and E4. As you can see, the way it only needs a change in one amino acid, in the case to be E4 instead of E3, since the change in the position 112. If you have the change in this amino acid, you will have different type of function of the protein. In the case of E4, you will have the LDL receptor binding protein in this region. And in the case of E3, the lipid binding region will change. And this change in the lipid binding region will confer to the E4 misfolding protein, in the case that this will confer the people to Alzheimer's disease a great risk factor for development of cognitive impairment. If we were trying to understand what means have at least one copy or two copies of E3 or E4, you can see it in this graph. If you have E3 and E3 genotyping, you have an average risk at, let's say, 1:1 to have a cognitive impairment. But if you have at least one copy or two copies of apolipoprotein E4, you will have 3.2 times more likely, or up to 14.9 times more likely to develop cognitive impairment. The biology of the apolipoprotein is very complex because in some point of the function, you will have gain of toxic function, but also you will have loss of physiological function. And this is when you have apolipoprotein E4 genotyping. You have more brain atrophy, more neural toxicity, more amyloid beta aggregation, more tangle formation, more aberrant brain activity. And in the other way, you will have less synaptic function, less glucose metabolism, less neurogenesis, less amyloid beta clearance, vascular function, mitochondrial function, and more important, you will have less lipid cholesterol metabolism inside the brain. So there are so many different pathways that ApoE can lead the development of Alzheimer's disease. In the case of ApoE, has been shown that has very important roles in metabolism, in synaptic plasticity, and transcription. If these pathways are impaired, for sure you will develop Alzheimer's disease. In the other way, if you have low amyloid beta clearance and high amyloid beta aggregation, you will have amyloid beta pathology that also can lead to Alzheimer's disease. And for the other end, you can also have impairment in the cerebral vasculature integrity. And this impairment can lead you to amyloid beta pathology and also to neuroinflammation. And this neuroinflammation has been shown that has a good amount of influence in development of Alzheimer's disease too. The way that we are measuring right now apolipoprotein is that we take blood sample from the participants, we perform a DNA extraction measurement, and we measure the concentration of DNA. Then you can set up the experiment to try to do the ApoE status. If each participant has genotyping for E2, E3, or E4, we do it by real-time PCR, and we obtain these different type of results. We will have a person that have E2 genotyping, E2, E3, and so on. And course, the one we are more interested in is to know which percentage of the population has E3, at least one copy or two copies of E4. And this is the first paper that we published about the characterization that Dr. Britton already talked a little bit about this. And I want to show you this graph because it shows the distribution of the ApoE genotypes in our cohort. We divided by group, the group of Alzheimer's disease, the group of mild cognitive impairment, or the group that has no cognitive impairment. What we show that is that the group of Alzheimer's disease, at least 57% of the participant has at least one copy. In the group of mild cognitive impairment, 50% of the participant has at least one copy of E4, and in the controls has 20%. There is a huge difference, and this is what we expect, that we expect to find that people with Alzheimer's disease and mild cognitive impairment have more ApoE E4 genotyping. This was already explained by Dr. Britton that when we perform logistical regression analysis of factors associated with cognitive impairment, we combine these two groups, and we found that the people that has at least one copy of E4 has more likelihood to have mild cognitive impairment. We in the last paper that we published that describes our cohort, that is the Panama Gene Research Initiative longitudinal study, we update and we include more samples of the distribution of the ApoE genotype by groups, but now we divide it by groups of age, 65 to 74, 74 to 84, and 84 and so on. So what we found that was that this is a graph just to show you the distribution of the ApoE, E3, and E4 genotyping. It's expected that this group is, and also the E2 are the one has less percentage of the population of genotyping of E4. Now, this in the last few years has been shown that also ApoE has been a target for gene therapy. What the people is trying to do is to trying to silence or converting the ApoE E4 gene to ApoE 3 or E2 by CRISPR. And why they want to do that is because they want to try to increase lipidation. They want to anti-ApoE immunotherapy. They want to try to correct the structure. They want to see if they can degrade this protein and treat also with ApoE E2, that has been shown that as a protective value when the people has this genotyping. And also has been shown that there are so many molecules that interact with E4 that can downstream signaling. You can try to crosstalk with amyloid beta. You can try to see if you can increase the lipidation, and also try to see which receptors can interact with ApoE to try to mimic the ApoE and try to diminish the related signaling of ApoE E4. The other set of biomarkers that we were trying to study in our protocol were these cerebrospinal fluid biomarkers. Why is that? Because it has been shown that it is like you are doing an autopsy of the brain. When you obtain this fluid, you are in touch with all the proteins that are in the brain. So it's an indirect way to see if the brain has plaques and has neurofibrillary tangles in the case of tau protein and in the case of amyloid beta. So we're trying to see if we can measure the amyloid beta proteins that are forming these amyloid plaques, and also we can measure also the tau proteins that are forming the tau protein clumps and disintegrating the microtubules in the neuron. What we did was, first, we look for the change in the levels of established CSF biomarkers in different settings for the nervous system disease. we look for Alzheimer's disease. What is happening with amyloid beta proteins in Alzheimer's disease? We are seeing that are lower. What we looking for tau protein that they are increased the levels, and also an increased level for phospho-tau. What happens in normal aging? You see no change in these protein levels. And also we were trying to track what is happening in vascular dementia, because we've been trying to relate which type of dementia besides Alzheimer's we can find in our populations. And we see that in the case of vascular dementia, you will see a similar pattern that for Alzheimer's disease, but only differs in the case of phospho-tau 181 that has no change. Here you can see that the different values in picograms per milliliter that you expect to find in the case for amyloid beta, for controls, for total tau and phospho-tau. So now we can compare with our results against these controls. What we did was, the first thing what we have to do was to standardize the detection of these three proteins in CSF. And we did by ELISA in different diagnostic groups: Alzheimer's, mild cognitive impairment, vascular dementia, and normal controls. What we found was that we have a different diagnostic accuracy for different biomarkers. We have an area under the curve. That means that we have a diagnostic accuracy, we have the cutoff value, and we have sensibility and specificity. We found that when we combine two proteins, a ratio that is made by amyloid beta and phosphorylated tau, we found the best combination, and we have at least 96% of diagnostic accuracy, 90% of sensibility, and 88% of specificity. This is the best combination for us to use as a biomarker in CSF when we are trying to differentiate between the controls and the Alzheimer's disease group. When we compare this ratio of phospho-tau amyloid beta in different groups, we found that we can not only differentiate Alzheimer's disease versus normal controls, that also we can differentiate mild cognitive impairment versus normal controls. And this is very important because, as you know, mild cognitive impairment can be a transition state for us, for so many people that go to Alzheimer's disease, but also there is a proportion of the population that can steadily stay in this group of mild cognitive impairment. It means that doesn't go to the group of Alzheimer's disease. And this is very important to characterize too. When we see the area under the curve, in terms of sensibility and specificity, we have 96.7%. That is very good when you have biomarkers. So far we have been talking that, and we have been shown that ApoE genotyping is a genetic risk factor for not only Alzheimer's disease and also for mild cognitive impairment. In our cohorts, those participant that has at least one copy has 5 times more likely to develop cognitive impairment. And of course, it's a feasible biomarker in clinical setting. That is very important. When you are trying to identify biomarkers that can be implemented in clinical. So also, we found that this amyloid beta, this CSF biomarker profile that has been shown in different studies that works very well for identifying also controls versus Alzheimer's disease. We also can use it in our population. Has an excellent diagnostic accuracy, and the best combination of biomarker is the one when you combine amyloid beta and phosphorylated tau. So what is the main challenge when you are trying to detect early Alzheimer's disease? Because so far we've been shown that when you have these proteins in the brain, you will have cognitive impairment, but we want to move backwards to try to see if we can define or we can obtain a biomarker in early phase of Alzheimer's disease. Most of the biomarkers so far has been present in this graph because you will have false negative and true and false positive. You have on There is a huge threshold right now that we want to divide it and obtain really good true negative that will be healthy age population, a true positive, the people that has Alzheimer's disease but in early stage. That's why we are looking blood-based biomarker for Alzheimer's disease. So in the hunt for blood-based biomarkers, you will find different biomarkers that right now we are looking for. For examples, we are trying to see if we can detect amyloid beta 142 and 140 in blood. That will indicate that you will have amyloid plaques in your brain. In the case that you can detect tau proteins and phosphorylated tau proteins in blood, that will also indicate you will have neurofibrillary tangles. But also we are trying to see if we can indicate inflammation, and you can do it by these proteins over here. And also, more important, you will try to identify if you have axonal injury. That is also can be led by inflammation in the brain. So this next slides will show you a summary of results. They are trying to investigate which plasma amyloid as a biomarker of cerebral amyloidosis, because this can be as the beginning of what we expect to find in later years for those people that has Alzheimer's disease. You have different methodologies that are very expensive, are very specific, as MALDI imaging, as spectral cytometry. And what you are doing right now is to try to see if you can correlate these blood-based biomarkers with imaging. And that's what you have here, receiver operation characteristic. When you have amyloid beta positive versus amyloid beta diagnosed by PET and CSF that has been shown and validated as biomarkers for amyloidosis. And here, in this case, you have a very good area under the curve, in the case that you related with amyloid beta, and in the case you related with CSF, 88% and 88 and 85%. And also here, you can see that there are other methods that are more easy to implement in clinical setting, as ELISA, ELIXIS, and SIMUA. These are equipment automatized that you can use in clinical setting that has lower diagnostic accuracy, but also you can use it very well to differentiate between the groups. So what we did is that we use a set of 30 proteins, and we did a multiplex immunoassay called human map. And we used 30 candidates marked to diagnose Alzheimer's disease. That is what we performed, and what I already told Dr. Gabrielle Britton about the Gini plot and the ranking of protein. And we did was a lot of cytokines, a lot of pro-inflammatory biomarkers, and other biomarkers that has hormone functions. And we published this paper called Serum-Based Protein Profile for Alzheimer's and Cognitive Impairment in the Early. So as Dr. Britton already mentioned, we use it as a combination of protein because it's too hard to differentiate one of another. But also we want to try to explore what is happening each by each biomarkers along with the groups. And we found that there is two biomarkers, IL-18 and I309, that we can use very well to differentiate within the groups. Here we see that the levels of IL-18 decrease in the group of Alzheimer's disease, and is significant. There is significant difference. And in the case of I309, the levels increase. What this served for us was to try to see how we can translate this information into basic research. What we are doing right now is trying to see cell cultures with I309 in axonal in neuron cells to see if we can stimulate and see if we found the same thing that we look in the blood. And this work is done by Dr. Carreira that was shown by Dr. Britton previously. So to end my presentation, I like to show this slide because it shows you different settings as blood biomarkers, structural MRI, and what shows you is that we are trying to move forward to see if we can implement blood-based biomarkers because it's high at the levels of effectiveness, time effectiveness, is low and it's not invasive and has a very high accessibility. It's an optimal first-line biomarker. Of course, if you have a very high negative predictive value, as we have mentioned before, you can rule out with these other three settings if you don't have any biomarkers for Alzheimer's disease. So I try to end my presentation with this message that we are moving forward and try to establish which biomarkers we can find in blood to try to be more effective in try to identify not only cytokines, but also genes that has maybe not high impact as the familial genes of Alzheimer's disease, but also we can use it as a biomarker discovery to try to find out which biomarkers in blood has very effectiveness in trying to identify this case of Alzheimer's disease. So our research is trying to move forward in the way that we identify what is the minimum sufficient evaluation using cognitive testing and also using biomarkers. Because we want to try to identify also possible interventions to set this, to try to clusters these participants in different groups in the way that we establish the infrastructure so we can participate as a multi-center clinical trials. Okay, because this is the way we are thinking to move in the forward and in the next future is to cluster these participants in the way that we are going to see, okay, this cluster of participants has this related gene. So now we can go to try to, if they can participate in a clinical trial that affect a specific pathways to involve this gene and this cytokine. So that's where we're moving right now, and we expect to find in the next few years. And of course, promote healthy aging is the very message for the people that work with us to see if we can identify these by risk factors, also try to establish a line of promoting healthy aging. Thank you very much to all these organizations. I work in the CASAT AIP, and of course, we need, there's so many people involved in this project, as Dr. Britton mentioned, and we are very glad to are here with you sharing our research. Thank you very much.
Thank you, Dr. Villarreal, for your nice lecture. And now we are going to have a short question answer session. So we have received more than 60 questions from different participants, but due to time, it may not be possible to have all questions here, but we will discuss some selected and relevant questions. So now I am inviting our secretary, organizing secretary, Dr. Vikas Kumar Roy, to chair this session.
Thank you, sir.
Thank you.
So Villarreal, one question is there. Is there any role of calorie restriction in preventing Alzheimer's disease?
I don't know if that question is for me, Gabi. Calorie restriction for Alzheimer's disease.
Okay.
I don't think that's been tested directly yet. I don't think there's been a formal. What we do know that it has been associated with better with longevity.
Yeah,
but not
it's
necessarily.
more related to aging, not to Alzheimer's disease. Yeah, calorie restriction patterns have been related to aging to improve longevity, but not related to Alzheimer's.
Okay. So next question is that what is the most challenging in terms of dealing with a patient undergoing Alzheimer's disease?
Well, the most challenge is to, right now we are moving forward in our research in the way that we are trying to identify those subjects or those people that are at risk for Alzheimer's disease. So because once you get the disease, it's very hard to work as a patient or as a subject of research because you will not give so many information and you will have already all the patterns, you will have all the proteins in your blood. And of course, you can easily detect and you will identify very clearly a people that has Alzheimer's disease. So what is really challenging right now is trying to identify those people that has mild cognitive impairment or those people that has subjective mild cognitive impairment. Those people that has memory complaints that you don't know if they will move forward for Alzheimer's disease or not. And that's why that's what is really challenging. And you will have to have a really good battery of cognitive testing and use of health of biomarkers to try to differentiate and try to cluster them in groups, because other way will be really difficult to see. And more important, you will have to conduct longitudinal clinical studies. You will have to follow them in time. And actually, something that we are discussing right now is one time you need to be between the visits. Some study has been shown that you can follow them by maybe 6 months, 1 year, 1 year and a half, and maybe so on. So that is the most challenging part, that you will have to keep them in the study and keep them happy to try to keep in your study. So it's really challenging actually to work with normal and possible mild cognitive impairment groups.
Okay, so next question is that what is the most prevalent serum marker for all the neurodegenerative disease? What is the most prevalent markers out of all the many markers? What is the most prevalent marker?
Well, the most prevalent marker so far has been amyloid beta and tau protein. That has been the central role of the whole amyloid beta hypothesis that has been challenged for I think the past 50 years so far. So there's so many researchers, and we have a couple months ago a conference with people from all Latin America, and we were discussing actually what is the central role of amyloid beta and if we need to move forward to tau pathology, because some research has been focused their attention that maybe amyloid beta is not the real deal about getting Alzheimer's disease. So that's something that has been discussed so far.
Okay, one very common question is being asked that Alzheimer's and Parkinson's and other neurodegenerative diseases, what is the chances of their inheritance from parental to the offspring? If parents have this kind of disease, what is the chances of getting in their offspring?
Only the statistics shows that between 1% to 5% of all Alzheimer's disease cases are hereditary. So, but what is happening if your parent have these genes, is very, very likely that you will have it and you will develop the disease. So has a huge effect size, these 3 genes, when you have it. But it's very unlikely that you will have it in the population. In Panama, so far, we are in our cohorts, we are not detecting any of them so far. So it's very, very uncommon that you will find Alzheimer's disease, familial Alzheimer's disease, and also Parkinson.
Okay. So now some questions are from my side because we are going to end this session. Like you have said, this apolipoprotein is elevated in terms of that dementia or Alzheimer's disease. So this apolipoprotein is expressed due to Alzheimer, or these proteins are expressed to counteract the challenges caused by the Alzheimer's? It is a
They
different.
are they are expressed as a normal. The thing is, has been shown that when you express a apolipoprotein E4, that you will have lipid transport impairment. But when you have the other genotyping, you don't have this problem. So all of us express apolipoprotein, but it will depend if you express E4 or other genotypes.
Okay. One more question that you have said that biomarkers. I have seen one protein is the adipokine as a biomarker.
Yeah.
So is there any other adipokine like visfatin, apelin are being developed as a biomarker for this neurodegenerative disease?
In our panel, no, we are not found any other adiponectin. Yeah.
Okay, now from previous session, still there are some questions for Dr. Britton. So I request Dr. Britton to answer a few questions. Okay, combined we can ask. At what age can we see the expression of interleukin in the blood?
At what age can you see the expression of what in the blood?
Interleukin, IL, interleukins.
Interleukins.
You have said
I think they're present at any age, right? I mean, you have
Yeah.
interleukins at any age. It's just that they're expressed at different levels. But yeah.
What is expected that when you have Alzheimer's, there is an impairment or the increase or decrease levels of these cytokines. But
Yes.
you will find it in any setting of life. Yeah.
Okay, another question is there any geographical influence on the biomarkers of Alzheimer's disease?
Geographical influence, no, but there appears to be an ethnic influence. In some biomarkers are expressed more in some ethnic groups than in others. So those differences have been found. For instance, those data are collected in the United States where they show differences between Hispanics or non-Hispanic whites and African Americans, and they've shown differences in the expression of biomarkers and in terms of their predictive value for Alzheimer's. Yes.
And next question is there any natural medicine through which we can prevent the disease of all this?
Natural medicine, probably the only one is like exercise, physical exercise, the best natural medicine.
Actually, that's the more common and the thing that has been shown in all research. Healthy living. You have to eat well. You have to do exercise. You have to keep low your carb. Everything has been shown the same thing. Healthy living.
Healthy lifestyle. Yep.
Healthy lifestyle. That's it.
Is there any report on the plant extract? Some bioactive chemicals from plant is being used as a treatment for
Like
curcumin and all. Do you have any idea?
Well, we have a strong group from Indicasat working with extract plants and what is the effect of for amyloid beta if we can block amyloid beta receptors. I think Dr. Sambasiva, that will be very nice if you can invite them to a webinar. They will explain. We have a very good scientist doing that research, and they will have a 2 hours talk about it for sure.
Yeah. In fact, they're doing structural modifications of a curcumin molecule. So trying to determine its effect on amyloid beta aggregation and other parameters. Yeah, very interesting.
I think Alcibiades, Marcelino and group, they are working on plant extracts and microbial extracts. No, that's what I was referring them.
Yeah,
Yes.
yeah. Right now, Johan, I think Johan go up a couple more.
Group is working on that. Marcelino group. They are extracting so many. They have developed so many plant extracts and microbial extracts. Micro and marine organisms also. They tried so many. That's what I'm just reminding Vikas to ask that Maybe you are aware of that. You can sell. You can tell about those products. Several. I remember Marcelino Group has done so many products which are positive for control of Alzheimer's and Parkinson. Am I right?
No, not specifically for Alzheimer's and Parkinson's. I mean, they're looking at plant extracts for a number of different things, but no, the only person who's working on something specific to Alzheimer's is Johan, who works in Marcelino's group.
Yes.
And she's doing the curcumin molecule, and they're doing different like structural modifications and testing against synthetic, doing different bioassays tests. Yes.
No, actually, because like Mizoram, Panama is also very rich in forest area. So many forest plant extract products actually they might have been tried in Panama Institute. That's what I was telling them. So that,
yes.
That may be one of the alternative for Mizoram also, because 90% of the Mizoram is also forest area. So
oh, okay. Interesting.
It is almost similar to Panama here. In dense forests are there. 90% forest area. So
okay.
we have very rich biodiversity here. Hotspots are there. So same, no, similar work. They are interested. That's why they are asking.
Okay. Yeah, it would be good for you to invite them on a webinar. They would be very excited to do that work. Yeah.
Thank you, Britton and Alcibiades.
Thank you. Thank you.
So now we are at the end of today's lecture. So we secretary for this event, and on behalf of my Vice Chancellor, Mizoram University, we are very thankful to two speakers and all participants who just saved time to attend this lecture. So I hope that these two lectures may be very useful. And now we are going to end this session. And I am again requesting participants, if they want a certificate, please communicate to us. We will provide
You can also
certificate.
tell the same collaboration. You can say tell with Britton.
Madam, just we want to have a collaboration with INDICAST for our teaching and research. On direction of our Vice Chancellor, we have already initiated process, but now we are admitting our PhD student, and we want to collaborate for PhD work, research work, through student participation, through teachers participation. We want to have INDICAST scientists to spend some time here in Mizoram University. So we are looking for this possibility. So
I'm sure that would be a great idea.
Yes, madam. Yes. Okay. So with these words, now we are going to close today's lecture. Again, I would like to thank our both distinguished speakers and participants. Thank you to everyone. Thank you.
Thank
Good
you.
night.
Bye-bye. Bye-bye.